The hippocampus, named after its resemblance to the seahorse, from the Greek pi kappa alpha mu pi omicron, seahorse from pi omicron hippos, horse, and kappa mu pi omicron campos, sea monster, is a major component of the brains of humans and other vertebrates. Humans and other mammals have two hippocampi, one in each side of the brain. The hippocampus belongs to the limbic system and plays important roles in the consolidation of information from short-term memory to long-term memory, and in spatial memory that enables navigation. The hippocampus is located under the cerebral cortex, allocortical, and in primates in the medial temporal lobe. It contains two main interlocking parts, the hippocampus proper, also called Ammons horn, and the dentate gyrus. In Alzheimer's disease, and other forms of dementia, the hippocampus is one of the first regions of the brain to suffer damage. Short-term memory loss and disorientation are included among the early symptoms. Damage to the hippocampus can also result from oxygen starvation, hypoxia, encephalitis, or medial temporal lobe epilepsy. People with extensive, bilateral hippocampal damage may experience anterograde amnesia, the inability to form and retain new memories. In rodents as model organisms, the hippocampus has been studied extensively as part of a brain system responsible for spatial memory and navigation. Many neurons in the rat and mouse hippocampus respond as place cells, that is, they fire bursts of action potentials when the animal passes through a specific part of its environment. Hippocampal place cells interact extensively with head direction cells, whose activity acts as an inertial compass, and conjecturally with grid cells in the neighboring entorhinal cortex. Since different neuronal cell types are neatly organized into layers in the hippocampus, it has frequently been used as a model system for studying neurophysiology. The form of neural plasticity known as long-term potentiation, LTP, was first discovered to occur in the hippocampus and has often been studied in this structure. LTP is widely believed to be one of the main neural mechanisms by which memories are stored in the brain. Name: the earliest description of the ridge running along the floor of the temporal horn of the lateral ventricle comes from the Venetian anatomist Julius Caesar Aranzi, 1587, who likened it first to a silkworm and then to a seahorse. Latin, hippocampus from Greek, pi omicron, horse, and kappa mu pi omicron, sea monster. The German anatomist Duvernois, 1729, the first to illustrate the structure, also wavered between seahorse and silkworm. Ram's horn was proposed by the Danish anatomist Jacob Winslow in 1732, and a decade later his fellow Parisian, the surgeon de Garanget, used Cornuamenes' horn of the ancient Egyptian god, Amun, who was often represented as having a ram's head. This has survived in abbreviated form as Ca in naming the subfields of the hippocampus. Another reference appeared with the term pes hippocampi, which may date back to Dimmerbroek in 1672, introducing a comparison with the shape of the folded back forelimbs and webbed feet of the mythological hippocampus, a sea monster with a horse's forequarters and a fish's tail. The hippocampus was then described as pes hippocampi major, with an adjacent bulge in the occipital horn, described as the pes hippocampi minor and later renamed as the calcar avis. The renaming of the hippocampus as hippocampus major, and the calcar avis as hippocampus minor, has been attributed to Felix Wieck Diazer systematizing nomenclature of parts of the brain in 1786. Mayer mistakenly used the term hippopotamus in 1779, and was followed by some other authors until Carl Friedrich Berdake resolved this error in 1829. In 1861 the hippocampus minor became the center of a dispute over human evolution between Thomas Henry Huxley and Richard Owen, satirist as the Great Hippocampus Question. The term hippocampus minor fell from use in anatomy textbooks, and was officially removed in the Nomina Anatomica of 1895. Today, the structure is just called the hippocampus, with the term cornu-amines surviving in the names of the hippocampal subfields Ca1-Ca4. The term limbic system was introduced in 1952 by Paul MacLean to describe the set of structures that line the edge of the cortex, Latin limbus meaning border. These include the hippocampus, cingulate cortex, olfactory cortex, and amygdala. Paul MacLean later suggested that the limbic structures comprise the neural basis of emotion. The hippocampus is anatomically connected to parts of the brain that are involved with emotional behavior: the septum, the hypothalamic mammillary body, and the anterior nuclear complex in the thalamus, and is generally accepted to be part of the limbic system. Anatomy. The hippocampus can be seen as a ridge of gray matter tissue, elevating from the floor of each lateral ventricle in the region of the inferior or temporal horn. This ridge can also be seen as an inward fold of the archicortex into the medial temporal lobe. The hippocampus can only be seen in dissections as it is concealed by the parahippocampal gyrus. The cortex thins from 6 layers to the 3 or 4 layers that make up the hippocampus. The term hippocampal formation is used to refer to the hippocampus proper and its related parts. However, there is no consensus as to what parts are included. Sometimes the hippocampus is said to include the dentate gyrus and the subiculum. Some references include the dentate gyrus and the subiculum in the hippocampal formation, and others also include the presubiculum, parasubiculum, and entorhinal cortex. The neural layout and pathways within the hippocampal formation are very similar in all mammals. The hippocampus, including the dentate gyrus, has the shape of a curved tube, which has been compared to a seahorse, and a ram's horn, cornu-amines. Its abbreviation Ca is used in naming the hippocampal subfields, Ca1, Ca2, Ca3, and Ca4. It can be distinguished as an area where the cortex narrows into a single layer of densely packed pyramidal neurons, which curl into a tight U shape. One edge of the U, Ca4, is embedded into the backward-facing, flexed dentate gyrus. The hippocampus is described as having an anterior and posterior part, in primates, or a ventral and dorsal part in other animals. Both parts are of similar composition but belong to different neural circuits. In the rat, the 2 hippocampi resemble a pair of bananas, joined at the stems by the commissure of fornix, also called the hippocampal commissure. In primates, the part of the hippocampus at the bottom, near the base of the temporal lobe, is much broader than the part at the top. This means that in cross-section the hippocampus can show a number of different shapes, depending on the angle and location of the cut. In a cross-section of the hippocampus, including the dentate gyrus, several layers will be shown. The dentate gyrus has 3 layers of cells, or 4 if the hilus is included. The layers are from the outer in, the molecular layer, the intermolecular layer, the granular layer, and the hilus. The Ca3 in the hippocampus proper has the following cell layers known as strata: lacunosummoleculare, radiatum, lucidum, pyramidal, and oriens. Ca2 and Ca1 also have 4 each of these layers and not the lucidum stratum. The input to the hippocampus, from varying cortical and subcortical structures, comes from the entorhinal cortex via the perforant path. The entorhinal cortex, EC, is strongly and reciprocally connected with many cortical and subcortical structures as well as with the brainstem. Different thalamic nuclei, from the anterior and midline groups, the medial septal nucleus, the supramammillary nucleus of the hypothalamus, and the raphe nuclei and locus coeruleus of the brainstem all send axons to the EC, so that it serves as the interface between the neocortex and the other connections, and the hippocampus. The EC is located in the parahippocampal gyrus, a cortical region adjacent to the hippocampus. This gyrus conceals the hippocampus. The parahippocampal gyrus also includes the perirhinal cortex, which plays an important role in the visual recognition of complex objects. There is also substantial evidence that it makes a contribution to memory, which can be distinguished from the contribution of the hippocampus. It is apparent that complete amnesia occurs only when both the hippocampus and the parahippocampus are damaged. Circuitry. The perforant path is the main output pathway of the entorhinal cortex, EC. The flow of information in the hippocampus is largely unidirectional. Axons from the pyramidal cells, in EC layer 2, perforate the subiculum to project mainly to the granular layer in the dentate gyrus, first synaptic connection. Some axons project to CA3 and a lesser number project to CA1. The dentate granule cell axons, called mossy fibers, pass on the information to the dendrites of CA3 pyramidal cells, second synaptic connection. From there, CA3 axons called Schaefer collaterals leave the deep part of the cell body and loop up to the apical dendrites and then extend to CA1. Axons from CA1 then project back to the entorhinal cortex, third synaptic connection, completing the Tri Synaptic loop or circuit. Basket cells in CA3 receive excitatory input from the pyramidal cells and then give an inhibitory feedback to the pyramidal cells. This recurrent inhibition is a simple feedback circuit that can dampen excitatory responses in the hippocampus. The pyramidal cells gives a recurrent excitation, which is an important mechanism found in some memory processing microcircuits. Several other connections play important roles in hippocampal function. Beyond the output to the EC, additional output pathways go to other cortical areas including the prefrontal cortex. A major output goes via the fornix to the lateral septal area and to the mammillary body of the hypothalamus, which the fornix interconnects with the hippocampus. The hippocampus receives modulatory input from the serotonin, norepinephrine, and dopamine systems, and from the nucleus reunions of the thalamus to field CA1. A very important projection comes from the medial septal nucleus, which sends cholinergic and gamma-aminobutyric acid, GABA, stimulating fibers, GABAergic fibers, to all parts of the hippocampus. The inputs from the medial septal nucleus play a key role in controlling the physiological state of the hippocampus. Destruction of this nucleus abolishes the hippocampal theta rhythm and severely impairs certain types of memory. Hippocampal regions. Areas of the hippocampus are shown to be functionally and anatomically distinct. The dorsal hippocampus, DH, ventral hippocampus, VH, and intermediate hippocampus serve different functions, project with differing pathways, and have varying degrees of place cells. The dorsal hippocampus serves for spatial memory, verbal memory, and learning of conceptual information. Using the radial arm maze, lesions in the DH were shown to cause spatial memory impairment while VH lesions did not. Its projecting pathways include the medial septal nucleus and supramammillary nucleus. The dorsal hippocampus also has more place cells than both the ventral and intermediate hippocampal regions. The intermediate hippocampus has overlapping characteristics with both the ventral and dorsal hippocampus. Using anterograde tracing methods, Chenkisksa and Swanson, 2007, located the moderate projections to two primary olfactory cortical areas and prelimbic areas of the medial prefrontal cortex. This region has the smallest number of place cells. The ventral hippocampus functions in fear conditioning and affective processes. Anagnosteras et al., 2002, showed that alterations to the ventral hippocampus reduced the amount of information sent to the amygdala by the dorsal and ventral hippocampus, consequently altering fear conditioning in rats. Historically, the earliest widely held hypothesis was that the hippocampus is involved in olfaction. This idea was cast into doubt by a series of anatomical studies that did not find any direct projections to the hippocampus from the olfactory bulb. However, Later work did confirm that the olfactory bulb does project into the ventral part of the lateral entorhinal cortex, and field CA1 in the ventral hippocampus sends axons to the main olfactory bulb, the anterior olfactory nucleus, and to the primary olfactory cortex. There continues to be some interest in hippocampal olfactory responses, in particular, the role of the hippocampus in memory for odors, but few specialists today believe that olfaction is its primary function. Theories of hippocampal functions. Over the years, three main ideas of hippocampal function have dominated the literature: response inhibition, episodic memory, and spatial cognition. The behavioral inhibition theory, caricatured by John O'Keefe and Lynn Nadel as slam on the brakes, was very popular up to the 1960s. It derived much of its justification from two observations: first, that animals with hippocampal damage tend to be hyperactive; second, that animals with hippocampal damage often have difficulty learning to inhibit responses that they have previously been taught, especially if the response requires remaining quiet, as in a passive avoidance test. Jeffrey Gray developed this line of thought into a full-fledged theory of the role of the hippocampus in anxiety. The inhibition theory is currently the least popular of the three. The second major line of thought relates the hippocampus to memory. Although it had historical precursors, this idea derived its main impetus from a famous report by William Beecher Scoville and Brenda Milner describing the results of surgical destruction of the hippocampi, in an attempt to relieve epileptic seizures, in Henry Molaison, known until his death in 2008 as patient HM. The unexpected outcome of the surgery was severe anterograde and partial retrograde amnesia. Molaison was unable to form new episodic memories after his surgery and could not remember any events that occurred just before his surgery, but he did retain memories of events that occurred many years earlier, extending back into his childhood. This case attracted such widespread professional interest that Molaison became the most intensively studied subject in medical history. In the ensuing years, other patients with similar levels of hippocampal damage and amnesia, caused by accident or disease, have also been studied, and thousands of experiments have studied the physiology of activity-driven changes in synaptic connections in the hippocampus. There is now universal agreement that the hippocampi play some sort of important role in memory. However, the precise nature of this role remains widely debated. The third important theory of hippocampal function relates the hippocampus to space. The spatial theory was originally championed by O'Keefe and Nadel, who were influenced by E. C. Tolman's theories about cognitive maps in humans and animals. O'Keefe and his student Dustrovsky in 1971 discovered neurons in the rat hippocampus that appeared to them to show activity related to the rat's location within its environment. Despite skepticism from other investigators, O'Keefe and his co-workers, especially Lynn Nadel, continued to investigate this question, in a line of work that eventually led to their very influential 1978 book The Hippocampus as a Cognitive Map. There is now almost universal agreement that hippocampal function plays an important role in spatial coding, but the details are widely debated. Later research has focused on trying to bridge the disconnect between the two main views of hippocampal function as being split between memory and spatial cognition. In some studies, these areas have been expanded to the point of near convergence. In an attempt to reconcile the two disparate views, it is suggested that a broader view of the hippocampal function is taken and seen to have a role that encompasses both the organization of experience, mental mapping, as per Tolman's original concept in 1948, and the directional behavior seen as being involved in all areas of cognition. So that the function of the hippocampus can be viewed as a broader system that incorporates both the memory and the spatial perspectives in its role that involves the use of a wide scope of cognitive maps. This relates to the purposive behaviorism born of Tolman's original goal of identifying the complex cognitive mechanisms and purposes that guided behavior. It has also been proposed that the spiking activity of hippocampal neurons is associated spatially, and it was suggested that the mechanisms of memory and planning both evolved from mechanisms of navigation and that their neuronal algorithms were basically the same. Many studies have made use of neuroimaging techniques such as functional magnetic resonance imaging, fMRI, and a functional role in approach avoidance conflict has been noted. The anterior hippocampus is seen to be involved in decision-making under approach avoidance conflict processing. It is suggested that the memory, spatial cognition, and conflict processing functions may be seen as working together and not mutually exclusive. Role in memory. Psychologists and neuroscientists generally agree that the hippocampus plays an important role in the formation of new memories about experienced events, episodic or autobiographical memory. Part of this function is hippocampal involvement in the detection of new events, places, and stimuli. Some researchers regard the hippocampus as part of a larger medial temporal lobe memory system responsible for general declarative memory, memories that can be explicitly verbalized. These would include, for example, memory for facts in addition to episodic memory. The hippocampus also encodes emotional context from the amygdala. This partly why returning to a location where an emotional event occurred may evoke that emotion. There is just a deep emotional connection between episodic memories and places. Due to bilateral symmetry, the brain has a hippocampus in each cerebral hemisphere. If damage to the hippocampus occurs in only one hemisphere, leaving the structure intact in the other hemisphere, the brain can retain near-normal memory functioning. Severe damage to the hippocampi in both hemispheres results in profound difficulties in forming new memories, anterograde amnesia, and often also affects memories formed before the damage occurred, retrograde amnesia. Although the retrograde effect normally extends many years back before the brain damage, in some cases older memories remain. This retention of older memories leads to the idea that consolidation over time involves the transfer of memories out of the hippocampus to other parts of the brain. Experiments using intrahippocampal transplantation of hippocampal cells in primates with neurotoxic lesions of the hippocampus have shown that the hippocampus is required for the formation and recall, but not the storage, of memories. Damage to the hippocampus does not affect some types of memory, such as the ability to learn new skills, playing a musical instrument or solving certain types of puzzles, for example. This fact suggests that such abilities depend on different types of memory, procedural memory, and different brain regions. Furthermore, amnesic patients frequently show implicit memory for experiences even in the absence of conscious knowledge. For example, patients asked to guess which of two faces they have seen most recently may give the correct answer most of the time in spite of stating that they have never seen either of the faces before. Some researchers distinguish between conscious recollection, which depends on the hippocampus, and familiarity, which depends on portions of the medial temporal lobe. Role in spatial memory and navigation. Studies on freely moving rats and mice have shown many hippocampal neurons to act as place cells that cluster in place fields, and these fire bursts of action potentials when the animal passes through a particular location. This place-related neural activity in the hippocampus has also been reported in monkeys that were moved around a room whilst in a restraint chair. However, the place cells may have fired in relation to where the monkey was looking rather than to its actual location in the room. Over many years, many studies have been carried out on place responses in rodents, that have given a large amount of information. Place cell responses are shown by pyramidal cells in the hippocampus and by granule cells in the dentate gyrus. Other cells in smaller proportion are inhibitory interneurons, and these often show place-related variations in their firing rate which are much weaker. There is little if any spatial topography in the representation. In general, cells lying next to each other in the hippocampus have uncorrelated spatial firing patterns. Place cells are typically almost silent when a rat is moving around outside the place field but reach sustained rates as high as 40 Hz, when the rat is near the center. Neural activity sampled from 30 to 40 randomly chosen place cells carries enough information to allow a rat's location to be reconstructed with high confidence. The size of place fields varies in a gradient along the length of the hippocampus, with cells at the dorsal end showing the smallest fields, cells near the center showing larger fields, and cells at the ventral tip showing fields that cover the entire environment. In some cases, the firing rate of hippocampal cells depends not only on place but also on the direction a rat is moving, the destination toward which it is traveling, or other task-related variables. In humans, cells with location-specific firing patterns have been reported during a study of patients with drug-resistant epilepsy. They were undergoing an invasive procedure to localize the source of their seizures, with a view to surgical resection. The patients had diagnostic electrodes implanted in their hippocampus and then used a computer to move around in a virtual reality town. Similar brain imaging studies in navigation have shown the hippocampus to be active. A study was carried out on taxi drivers. London's black cab drivers need to learn the locations of a large number of places and the fastest routes between them in order to pass a strict test known as the knowledge in order to gain a license to operate. A study showed that the posterior part of the hippocampus is larger in these drivers than in the general public, and that a positive correlation exists between the length of time served as a driver and the increase in the volume of this part. It was also found the total volume of the hippocampus was unchanged, as the increase seen in the posterior part was made at the expense of the anterior part, which showed a relative decrease in size. There have been no reported adverse effects from this disparity in hippocampal proportions. There are several navigational cells in the brain that are either in the hippocampus itself or are strongly connected to it, such as the speed cells present in the medial entorhinal cortex. Together these cells form a network that serves as spatial memory. The first of such cells discovered in the 1970s were the place cells, which led to the idea of the hippocampus acting to give a neural representation of the environment in a cognitive map. When the hippocampus is dysfunctional, orientation is affected, people may have difficulty in remembering how they arrived at a location and how to proceed further. Getting lost is a common symptom of amnesia. Studies with animals have shown that an intact hippocampus is required for initial learning and long-term retention of some spatial memory tasks, in particular ones that require finding the way to a hidden goal. Other cells have been discovered since the finding of the place cells in the rodent brain that are either in the hippocampus or the entorhinal cortex. These have been assigned as head direction cells, grid cells, and boundary cells. Speed cells are thought to provide input to the hippocampal grid cells. Role in approach avoidance conflict processing. Approach avoidance conflict happens when a situation is presented that can either be rewarding or punishing, and the ensuing decision-making has been associated with anxiety. fMRI findings from studies in approach avoidance decision-making found evidence for a functional role that is not explained by either long-term memory or spatial cognition. Overall findings showed that the anterior hippocampus is sensitive to conflict, and that it may be part of a larger cortical and subcortical network seen to be important in decision-making in uncertain conditions. A review makes reference to a number of studies that show the involvement of the hippocampus in conflict tasks. The authors suggest that a challenge is to understand how conflict processing relates to the functions of spatial navigation and memory and how all of these functions need not be mutually exclusive. Physiology. The hippocampus shows two major modes of activity, each associated with a distinct pattern of neural population activity and waves of electrical activity as measured by an electroencephalogram, EEG. These modes are named after the EEG patterns associated with them, theta and large irregular activity, LIA. The main characteristics described below are for the rat, which is the animal most extensively studied. The theta mode appears during states of active, alert behavior, especially locomotion, and also during REM, dreaming, sleep. In the theta mode, the EEG is dominated by large regular waves with a frequency range of 6 to 9 Hz, and the main groups of hippocampal neurons, pyramidal cells and granule cells, show sparse population activity, which means that in any short time interval, the great majority of cells are silent, while the small remaining fraction fire at relatively high rates, up to 50 spikes in 1 second for the most active of them. An active cell typically stays active for half a second to a few seconds. As the rat behaves, the active cells fall silent and new cells become active, but the overall percentage of active cells remains more or less constant. In many situations, cell activity is determined largely by the spatial location of the animal, but other behavioral variables also clearly influence it. The Theta mode appears during slow wave, non-dreaming, sleep, and also during states of waking immobility such as resting or eating. In the Theta mode, the EEG is dominated by sharp waves that are randomly timed large deflections of the EEG signal lasting for 25–50 milliseconds. Sharp waves are frequently generated in sets, with sets containing up to 5 or more individual sharp waves and lasting up to 500 milliseconds. The spiking activity of neurons within the hippocampus is highly correlated with sharp wave activity. Most neurons decrease their firing rate between sharp waves, however, during a sharp wave, there is a dramatic increase in firing rate in up to 10% of the hippocampal population. These two hippocampal activity modes can be seen in primates as well as rats, with the exception that it has been difficult to see robust theta rhythmicity in the primate hippocampus. There are, however, qualitatively similar sharp waves and similar state-dependent changes in neural population activity. Theta rhythm. Because of its densely packed neural layers, of all the brain structures which generate the hippocampal theta rhythm, the hippocampus generates some of the largest EEG signals as theta waves. In some situations the EEG is dominated by regular waves at 3 to 10 Hz, often continuing for many seconds. These reflect subthreshold membrane potentials and strongly modulate the spiking of hippocampal neurons and synchronize across the hippocampus in a traveling wave pattern. The Tri Synaptic Circuit is a relay of neurotransmission in the hippocampus that interacts with many brain regions. From rodent studies it has been proposed that the Tri Synaptic Circuit generates the hippocampal theta rhythm. Theta rhythmicity is very obvious in rabbits and rodents and also clearly present in cats and dogs. Whether theta can be seen in primates is not yet clear. In rats, the animals that have been the most extensively studied, theta is seen mainly in two conditions: first, when an animal is walking or in some other way actively interacting with its surroundings; second, during REM sleep. The function of theta has not yet been convincingly explained, although numerous theories have been proposed. The most popular hypothesis has been to relate it to learning and memory. An example would be the phase with which theta rhythms, at the time of stimulation of a neuron, shape the effect of that stimulation upon its synapses. What is meant here is that theta rhythms may affect those aspects of learning and memory that are dependent upon synaptic plasticity. It is well established that lesions of the medial septum, the central node of the theta system, cause severe disruptions of memory. However, the medial septum is more than just the controller of theta, it is also the main source of cholinergic projections to the hippocampus. It has not been established that septal lesions exert their effects specifically by eliminating the theta rhythm. Sharp waves. During sleep or during resting, when an animal is not engaged with its surroundings, the hippocampal EEG shows a pattern of irregular slow waves, somewhat larger in amplitude than theta waves. This pattern is occasionally interrupted by large surges called sharp waves. These events are associated with bursts of spike activity lasting 50 to 100 milliseconds in pyramidal cells of CA3 and CA1.
Please subscribe and thanks for watching.